Hello, everyone. I'm Garrett Weinzierl, and my guest today is a professor at the Department of Plant and Environmental Sciences at NMSU. Dr. Champa, thank you so much for joining me today. So how are you doing today? Doing well?
Good, very good.
Good. It's always early when we're recording here. So we are here today to talk about a new product, something that you have researched and come up with. So what is your product?
Our product is, we have produced an alfalfa line with the transgene, and I'll talk about in a minute. The plants grow a lot bigger, and so the growth rate has increased tremendously. So, for example, in a whole growing season, if, for example, the farmer harvests normal alfalfa maybe 5 times, with our product, they can harvest 7 times, which means we have really increased the biomass. Not only have we increased the biomass, we have also increased the nitrogen content of these plants. And this is by actually over-expressing, how I should say, is we are making a particular protein at very high levels. And this we have done using genetic engineering tools. And which I'm, if you, just to give you a simple background, and when I say genetic engineering, it's different from breeding, plant breeding, in the sense that we can take a gene from any living system and introduce, and after some manipulations in the lab, we can introduce it into a plant and express and expect it to show the characteristics that that gene is responsible for. So we took a gene from maize, from corn, and we introduced it into alfalfa. And this gene is responsible for making what we call a sugar, a sucrose. And sucrose, if you don't know, if you're not in the field, is the most important molecule in plants because it's the source of energy. And so that's the fuel that keeps the whole living system going, a plant system going. And so in our research, one of the most important things that people think of is to improve the rate of photosynthesis, because that's capturing light and converting it into chemical energy. So because that's a much more difficult process, even though many scientists are involved, our next step was to go and use what photosynthesis produces and convert it into sucrose, that sugar, which then goes to the rest of the plant and gives it the energy. So this particular step by which we can convert the first product of photosynthesis into sucrose is a particular enzyme, or what we call a catalyst, which is and the gene that's responsible for it, I'll just talk about it as SPS, or sucrose phosphate synthase. Okay, so we have taken a gene for SPS from corn and introduced it into alfalfa using genetic engineering tools. And this gene, and the reason we can do this in plants is because in plants, unlike animal system, we can take any cell from a plant and we can make it grow into a whole plant. So we introduce this gene into a single cell, and then we give it the right environment for the single cell to grow into a whole plant. And once the gene is introduced into one cell, the whole plant, every cell will have that gene, and it will be passed on to next generations like any other genes. So if you see our plants, we looked at the protein content in the aerial parts which the animals feed on. It's very high in proteins, high in amino acids. And remember, alfalfa is a legume. A legume is a plant where soybeans, beans all belong to this group of plants. They do not need nitrogen fertilizers. Because if you think about plants and about nitrogen, nitrogen is a very, very important molecule for a plant. Nitrogen and carbon are the two most important elements that the plant needs. So nitrogen is usually provided in farms and all by dumping nitrogen fertilizers in the soil. Alfalfa, like other legumes, do not require this nitrogen because they can form a symbiotic relationship with the bacteria in the soil, which will, and they form a unique structures, the organs, which we call the root nodule. This root nodule is filled with this bacteria, which can capture the atmospheric nitrogen and convert it into a form that plants can use. Just to remind you, the atmosphere has more than 75% nitrogen, but plants cannot use it. So it's only this bacteria that's formed this nodule structure can take the atmospheric nitrogen and convert it to a form that plants can use. So our goal is to improve the function of this nodule, because our goal is to make this plant be able to fix more atmospheric nitrogen and then convert it into a form that plants can use.
So is that why you landed on alfalfa? Because that was kind of my follow-up is why alfalfa? Why this specific plant?
Okay. The system that we have developed will work for any legume. So, which means I can use the same thing for peanut or for soybeans, and the same thing should work because the main focus is the root nodule. If I can improve the function of the root nodule, which means if I can capture more nitrogen, and if I can convert this nitrogen into a form that plants can use, I'm providing them with the nutrients that farmers do by dumping nitrogen fertilizers. So for the nitrogen to be, so the first thing is we have to capture the nitrogen from the environment. The bacteria does that. The bacteria will convert it into what we call ammonia. Now, what the plant does is to take this ammonia and using a plant enzyme converts it into what we call glutamine. I will not talk too much about all these things, but glutamine is important. The enzyme that converts ammonia that's produced by the bacteria is converted into glutamine by an enzyme which we call glutamine synthetase, or GS. So remember, I'm going to focus on SPS, which is in the carbon pathway, and GS, which is in the nitrogen pathway. So these are the two key enzymes that will allow the plant to take this nitrogen and convert it into a form that plants will use for everything else. If you think of all the macromolecules that make up a living cell, DNA, protein, RNA, they all require nitrogen and carbon. So if we can improve the amount of nitrogen that the plant can take and be able to utilize it, and to utilize nitrogen, we also need carbon, we need energy that the plant produces. So we call this whole process symbiosis because the bacteria provides nitrogen, the plant provides carbon. Okay, so this system, so what we have done by manipulating the expression of this gene, the carbon gene, we have increased the amount of sucrose that's going into the root nodule. When you have more sucrose, there is more energy to convert the ammonia that the bacteria has produced into glutamine. Glutamine is the starting point. This is what goes up into the plant, to all the parts of the plant, and starts giving the basic material for the synthesis of all these macromolecules. They turn on some signals that tells the plant you have to grow faster. So many genes are turned on, many genes are expressed, and they are responsible for plant growth. So for a farmer, if they can get 7 harvests of the forage in one growing season instead of 5, they're making a profit. And the quality of the forage is very good because it's very high in protein. Then the next thing is the same system that we have developed for alfalfa, we can use it for other legumes. And in other legumes, another important thing is besides the forage, the leaves, they also have fruits and seeds. So our system should also help in increasing the seed yield of legumes. If we can increase the amount of nitrogen that we can capture from the environment, from the atmosphere, and convert it into a usable form, and that we can do by increasing this protein that we call SPS, which is responsible for taking up photosynthate and converting it into sucrose.
So who is what, now that you have your kind of your final alfalfa product, who is your ideal customer?
Mostly dairy farmers, industry. Like, for example, the type of people we can go to is forage genetics. They are the biggest forage producers. We can go to any of the biotech companies, the plant agricultural biotech companies, like even Monsanto, DuPont, we can go to them. And all these, and since alfalfa has many recombinant DNA recombinant DNA technology has been applied to alfalfa, we don't even have to go through a lot of regulations because it's already there. But it's mostly biotech companies, and then, of course, the farmers eventually.
Okay. So, and I would assume at this point, is it patented?
Yes.
What was the patent process like? Can you talk about that?
I'll just tell you, Arrowhead did most of it, So I know we wrote a lot of stuff and there were lawyers, and I don't know those things, but the process was done by Arrowhead.
Did they make it easy for you, I'm assuming?
Yeah. That's why I don't know anything. Okay. I told them what I have done, and they asked me for all the technical details and what they did. And then I talked a lot to the patent lawyer. So yes, that's the process. I don't know much. See, as a scientist, we don't really worry that much about, we are just more excited about the research. So.
No, it comes across clearly. It's something you're very passionate about.
So you have, you can ask me any questions.
So what's next now that you've successfully you have this alfalfa that yields more harvest per year? What is next? You talked about possible other applications for similar plants.
Or to other legumes. Say, for example, we went to peanut. I'm just giving you an example.
Right.
So, in peanut, if we do this, we could increase the protein content in the seeds which we eat. Not only can we do that, we can increase the amount of fruits that we make. In alfalfa, we don't worry about the fruits. We're only interested in forage. But in other legumes, we can increase the yields. For example, soybean, which is a very important crop. So I think what we would do next is to go into another legume.
Okay.
Alfalfa, we still have lots to do. It's to improve the quality of the forage. So there are lots of things that we can do by using genetic engineering tools by which we can vary the quality of the protein that animals get from the forage, from the leaves.
Wonderful.
This technology can be used for many other plants.
Did you have a history with this type of research before? Because I took a look at your resume. You have a long resume. You've accomplished a lot so far.
I was working with a biotech company, and we were the first people in the field to have introduced a foreign gene into a plant and then see the effects of that.
Wow.
So that was in 1983. Okay. So it's a long time back.
You've been working on this for quite a while.
Yeah. Genetic engineering is what I do, but I have many other goals. We work a lot on chili plants. In chili, we are trying to make them resistant to a pathogen. So again, it's all genetic engineering tools. We have done a lot of work in soybeans. And again, so there are different projects that we have worked on. We have tried to improve the nutritional quality of legumes. So legumes are like alfalfa, soybean, they're legumes. And cereals are like rice. And so alfalfa, legumes are poor in methionine. Methionine is soy, I should be saying. It's not balanced completely. And so are the cereals, but they are both deficient in a particular amino acid. So that's why when you will see when we eat, we always have a cereal and a legume, beans and rice, tortilla and beans. So because alfalfa and legumes are poor in a particular amino acid, and we get that from cereals. So we have done genetic engineering by which we have increased the amount of this particular amino acid. And so the protein in this legume now becomes more balanced. So in alfalfa, besides, so I talked about improving the forage quality in alfalfa. We are also trying to see if we can increase both carbon and nitrogen. And so we have now, what we are doing, we are manipulating both the steps. And if we can take this technology to other legumes, I know we will have a huge impact. And when we talk about other legumes, we are talking about mostly legumes that have fruit that gives that the main part of that plant is the seeds, for example, lentils and beans. And so this what we are working on is something that can be taken to really poor countries where they need protein, and legumes are a source of proteins. So
if you can increase how often you can harvest, that's
for alfalfa we talked about,
right?
Just forage.
Exactly. So it's just
and soybeans will increase the amount of the beans. And if we went to something else, we can increase both the forage and also the fruits, because eventually the seeds, all the nutrients are going.
How long did it take from kind of day one on your research into alfalfa until you were at the end of your research and you were, getting it patented?
To the research, okay, so it's going on. So I would say we started working on this project about, I'm just trying to think, my students who started all this, maybe 10 years.
10 years, so a full decade. And you sound so passionate. In 10 years, for most people, for your general person, that seems like a long time to work on one thing. Is it difficult to stay to keep your eye on the target?
One thing leads to the next question and the next question. So yeah, I do this work, but I start with basic research. So since I work with students, they all, we get some result, we think of the next experiment, and that's what you give a student to do. And so even though this was one of the goals, but we went in many different directions to do basic research to eventually help with the main project. So, so it's both basic and applied. Eventually, of course, for the purpose of this research that we want to take to the real world, we did more applications, but we started as a basic research project, which really involves isolating the gene, doing all kinds of genetic engineering to make it possible for us to introduce it into a plant. See, we are doing genetic engineering, which is not an easy thing, but we can do it in plants more easily than in animals, because we have to introduce a gene into one cell, and then we can make the cell grow into a whole plant. And we make use of a bacteria that causes what we call tumors in plants. And we have engineered this bacteria such that we have removed all the tumor genes, but we have left enough of the genetic material that will allow this bacteria to take the foreign gene and put it into the DNA of that particular plant. And then from one cell where we have introduced our gene of interest, we grow it into a whole plant. That whole process of taking a foreign DNA, integrating it into the plant's DNA, and making it grow into a whole plant is itself a big thing that we have to develop. So for alfalfa, we spent quite a bit of time to make it an efficient system. Then we have to go for the right gene. We have to find a system for where we can isolate the gene. It's become a lot more easy now because we can synthesize genes if we know the DNA sequence. So we are doing that a lot in our research now. So yes, it's really in some ways taking DNA and trying to introduce it into a plant, and it should be such that the DNA should be able to do what it is supposed to do. It should make a protein. It should give you the particular trait that we are looking for. So,
and you have those consistent results more often than not.
Oh yeah. See, once your system is developed, it should work all the time. If it doesn't work, it means the students have made a mistake somewhere. So you go back and try to figure out what mistake they made. Say, for example, I'll just give you a simple explanation. if we work with young people, and say they're supposed to put a particular gene into a cell, and we work with many genes. I'm just giving you one example, and they made a mistake and put a wrong gene in, and then you're not seeing what you should see. Then you start again at ground zero to figure out why isn't it showing the trait. And then you figure out the student didn't put the right gene. Because see, in our lab, we work with many different genes. So I'm just talking about what we have done to produce the results that Arrowhead has been interested in. So if you go into my freezer, you'll find all kinds of DNA, which says this gene, that gene. So
it's all very organized. Yes, it sounds quite unforgiving. So it's very easy to find out when a mistake has been made.
Yeah. But you have to remember, to produce a whole plant, starting with when we start taking cells and introducing the gene, takes 9 months. So it's a long process. And so you've made a mistake, you've wasted so much time.
Yes, that's time you're not going to get back.
Having to check with the students, but there are ways we can check at every step,
Right, absolutely. Well, Dr. Champa, any closing thoughts before we end the interview?
Yeah, I would like to say that people have this misconception of genetic engineering. They're all afraid of GMOs. But GMOs is nothing different from what's happening in nature, okay? Yes, breeders are doing the same thing, but breeders have very limited tools. We have a lot of tools. And breeders have very few genes to play with because they have to do that between related plants. For us, the whole living system is a pool of genes. And if we do it in the right way, I don't know why it bothers the world so much. And I really want to put in this pitch that even breeders do some of the things that we do, but ours is so much more directed. It's under so many a big regulatory umbrella, whereas breeding is not. So in breeding, many times breeders have to go to wild plants, related plants, to bring the gene of interest by sexual crossing. And that's very dangerous because when you do just breeding, you're not just introducing your gene of interest, you're introducing also the DNA that's next to your gene. And you don't know what that DNA has. So
it's much less controlled.
Yeah, but what I'm saying is it takes a long time, but you still are not completely directed to one piece of DNA. You can bring DNA from all over the plant, and you're still trying to clean it up, but you cannot be 100% sure that you have gotten rid of everything else except your gene of interest. Whereas what we do is very directed. We get our gene, and we put it into a plant. And the whole living world is a source of genes for us. It's not just a related plant. We can go to bacteria, we can go to animal systems, anything we can do. But of course, plant people mostly focus on either plant genes or bacterial genes. So I think it opens up, and it's very efficient. It's like a one-step thing. You introduce the gene and that's done. Whereas breeding, you have to keep doing it over and over again to get a pure line. So I think we have a lot more tools. Like we are working on a project in our lab, which is we want to make chili plants resistant to a particular pathogen. The breeders have tried and it hasn't worked. So what we have gone into wild potato, which is resistant to this particular pathogen. And we have brought that gene, I've now introduced it into chili. And these chili plants are now resistant to this pathogen, which breeders have not been able to do just by breeding. So we have a lot more powerful tools in our hands. And so sometimes it is really trying to educate the people that there is nothing that we are doing that is any different from breeding. Ours is a much more efficient system, and we have opened up the whole living system as a source of genes.
Right. You even said we have more tools, and that's exactly what it sounds like. It sounds like it's a larger tool set.
Larger tool set. That's it, And we can really go for many different traits that breeders cannot do, we can do those things. And the breeders accept that what we are doing is something they cannot do. But we have to convince people who are not, well acquainted with this field, that there is nothing that we are doing that's dangerous. Or because you go into organic food stores, they'll say no GMOs. But I don't know whether they even understand what it means to be GMOs,
Right.
genetically modified organisms. And that's happening in nature without our help, so.
Right. Well,
Yeah.
Dr. Champa, thank you so much for sharing all of this with me today. Thank you for your time.
Okay, thank you. And if you have any questions, I hope I wasn't too technical. I tried not to be, but sometimes you deal with students and you become like that.
Well, I'm definitely a layman in this field, and I feel I understand more than I did going into the interview. So thank you.
Thank you.
Have a good one.
You too.